First panel this afternoon is a panel on CMC commercialization, and we're delighted to have Aine Henley and Peter Marks moderate this panel. I'll turn it over to them to introduce or have them self-introduce their panelists.
Thank you, Goudar. Good afternoon, everybody. We're first slot after the break. I don't know whether that's good or bad, but welcome to everybody, and thank you to the organizing committee for including a topic on CMC for this particular event today. It's one of those areas where people often wonder, what is CMC? Is it just manufacturing? Is it just supply chain? But CMC is that interface with research and the discovery of molecules or early scientific knowledge and the translation of that knowledge into medicines. And more specifically, it's the design and of processes, it's the understanding of attributes, it's the sourcing of materials, it's the scale up, the tech transfer, and ultimately supply to patients. So it's an intricate partnership of multiple disciplines across what is often called a tech ops organization. And I'm joined here today by Peter Marks, who's my co-partner for this session. And Peter doesn't need any introductions, but we really are excited to work together on this particular topic. And in addition, we have a number of scientists, leaders, industry veterans here, actually, from small companies, large companies, even academic institutions. So I'm going to actually hand over and have everybody introduce themselves briefly, and then we're going to get into a little bit of the discussion on CMC and how can we accelerate the advancement of medicines to patients. And in fact, that's why we're all here today. These medicines that are being discovered across the globe have no doubt got their journey through this path, but one which we feel that we can do even better, more efficiently, whilst at the same time ensuring patient safety and efficacy of these medicines. So let me hand over to Peter.
Yeah, so I'll just, so thanks for, and I have to thank you for doing a lot of the heavy lifting, getting things together. I think this is, a wonderful opportunity to have a discussion about something that is really critical, because it's, I think, in the academic world is where we have a lot of innovation coming from, but unless we can translate that innovation, for instance, in CRISPR, into products that are able to be made reproducibly and in an efficient manner such that the cost can ultimately be contained, we're going to have real issues. And so there's I think a lot of progress has been made to date, but there's still a long way to go. And I think the panel here, that'll be a nice time for discussion. So I'll pass it along. Chetan, go ahead.
Thank you, Peter. Thank you, Aine. Hi, I'm Chetan Goudar, and I come to you from the process development function at Amgen. And before that, I worked across the Bay at Bayer Healthcare, also in process development. And a big part of my experience has been in multiple marketing applications as well as IND filings, which really is where all the CMC dimensions do come together.
Hi, thank you for inviting me. Is the mic good? Pleased to be here. Oh, there we go. Pleased to be here. My name's Lynne Krummen, and I am presently head of product development at Biotechnology, which is actually just across our offices are just across the street here. And in that function is regulatory and program leadership, et cetera. Prior to joining Vir, which I've been at Vir for about 5 years, I was at Genentech and Roche for close to 30 years, doing a lot of activities in the technical development arena, CMC regulatory and product development.
Hi, I'm Alison Moore. I actually worked with Lynne many years ago at Genentech Roche. Then I spent 20 years at Amgen in different kinds of operational roles. And then until recently, I spent the last 5 years as Chief Technology Officer at Allogene Therapeutics, an allogeneic CAR-T company. Glad to be here.
I'm like they say on Sesame Street, I'm the one who's not like the other. I'm a professor of pediatrics at Stanford, but been involved in development of CRISPR-based gene editing for a number of years, but have been involved in the founding of several different CRISPR-based gene editing biotechs.
Hi, everybody. My name is Noemi Romero, and I am today the Global Supply Chain Head for Eli Lilly. Super happy to be here, not only because it's my first time at the CERSI Summit, and I've been very interested in all the topics this morning, but because I come from Indianapolis, Indiana, and the weather here is fantastic. I know many of you think it's cold. It is not cold. Thank you for inviting me, Anya and Peter.
Thank you, everybody. So let's kick off. I think we can all agree that actually the COVID pandemic had some silver linings. And one of those silver linings was the fact that we learned how to move efficiently, we learned how to partner, we learned how to leverage platforms. When we speak about platforms in the CMC world, by the way, we speak about standard approaches. We also learned through the COVID pandemic that ultimately working and partnering with the FDA or whatever agency we were launching our medicines in was highly beneficial. So I'm actually going to invite Lynne to give us some specific examples, given that Vir worked specifically on a monoclonal antibody therapy for COVID treatment. So Lynne, over to you to maybe kick us off on this theme.
Happy to, and to share some of the experiences that we had at Vir in developing sotrovimab, which was a monoclonal developed for the treatment of COVID-19 and was given an emergency use authorization early in 2021. And the reason I want to talk about sotrovimab and the sort of process that we use there is I think it's an excellent example of how really decades of monoclonal antibody process development and product understanding built a platform that at the moment of the emergency, we were able to leverage that information not only to streamline data collection, but also to accelerate development. And of course, we always want to say that, that platform of information gives us a good basis to think about risk-based decisions. It doesn't mean that there is zero risk, but I think that depth of understanding and breadth of understanding that had been developed in the industry with monoclonal antibodies really allowed for a catalyst of how to move very rapidly through development. And just for a sort of benchmark, the time from discovery of a DNA sequence till you actually have clinical trial material that can go into the clinic is roughly a year, and people will say, well, I can do it in 10 months. Some people will do it in 18 months, and that largely depends upon how the company is set up and how its manufacturing occurs, et cetera. For sotrovimab, that time frame was 5 months. So clearly we were leveraging information and making risk-based decisions that allowed us to accelerate quite a bit. And in fact, the EUA was issued 8 months after entry into the clinic. And so while we'll talk a lot about CMC acceleration today and how platforms benefit that, it was actually a very collaborative effort across many of the different divisions and departments within the agency that helped us to overall accelerate those timelines. So I wanted to mention there's a new guidance that just came out right before the holidays, which talks about accelerating development for COVID mAbs. And it really articulates a lot of the opportunities that were identified during the original portions of the pandemic and put those in guidance. And I think it's an early example, and I don't want to say the first example, but where we're really getting guidance on what types of platform information could be leveraged in certain circumstances. And I want to stress that this guidance is about monoclonals for COVID and in fact about specific types of monoclonals and molecules. But I think it's an excellent representation of the partnership that has developed between industry and the agency in terms of thinking about how can you prospectively plan to utilize some platform information to accelerate things. Obviously, things are always a case-by-case basis, and I really advise to try to take advantage of early collaborations to talk about the opportunities, but I think that new guidance is really relevant. The goal of our session today, though, is not to talk about the monoclonal antibody platform, but really to learn from how that platform was developed, what opportunities it gave us, and then talk about how can we think about building platforms that can be useful for more novel, innovative therapies, and really try to accelerate the building of platforms that I think across industry and with regulatory collaboration can build the kind of knowledge base that you need to be able to be more confident in your risk-based decision making. For this audience, I think it's important to note that the building of a platform for monoclonal antibody production probably took place over several decades. I'm going to say at least 3 decades, maybe more. And it started really with recognizing that there were certain unit operations that might be product independent, that could be characterized in a more modular way, and trying to collect enough data across as many processes as possible to then leverage that for new molecules going forward. I think over time, the bigger companies probably kind of led the way there. They had the biggest experiential databases and work with the agency to really talk about how could you leverage that information. But over that time, I think there's been a convergence in terms of publicly available and acknowledged characteristics of monoclonal antibodies that are important to how those molecules perform in the clinic, how to characterize them, and what parts of the process really impact those critical quality attributes and could impact product performance. And so when the emergency occurred, there had been this long build of experience that allowed us to really converge on a development strategy that leveraged a lot of the things that had been, I think maybe implemented on case-by-case basis, but now became part of a more practical, I think, exercise, and how can this all be leveraged to really speed development. For the molecule that we developed, I would say there's probably 5 main things that had to transpire to support the acceleration. The first was around the speed of introduction to the clinic. We really needed to think about sequentiality of things that we did and to feel comfortable and talk with regulators about how could we move forward, maybe without as complete of data packages that we might have had in sort of standard development, but also how are we going to manage that risk. We went from a product that was intended for IV use and only had a low concentration. We went through several iterations of higher concentration formulations, went through several scale changes, went through several manufacturing changes. And a lot of that was enabled because we could do a relatively strong comparability exercise as we went forward and really be able to implement those changes very rapidly. Another thing that was really important was being able to leverage shelf life information from early process versions to late process versions, understanding that by the time we were in the clinic, we had 1 month of stability information, and yet needed to have some extrapolation. So some of that was based on just historical knowledge of protein chemistry, degradation pathways, et cetera, but also thinking about how could we leverage that as we go forward to get closer to a commercial shelf life. So from a product perspective, it was really understanding and being able to characterize the antibody. From a process perspective, it was built on a foundation of what parts of the process impact product quality. And then there was a whole host of other things, which I'll pass on to others. about the manufacturing environment, about the supply chain that got built, et cetera, in a very rapid time frame.
Thanks, Lynne. And maybe we could invite Noemi to talk about supply chain. I know, Kathy, when you and I spoke initially about this topic, supply chain was one of those areas that you felt would be really helpful to understand a little bit more deeply, and no one better than Noemi to give us not just the Lilly experience during COVID, but just in general, how do we improve resilience of supply chains, and how do we ensure that they're not the bottleneck when it comes to enabling accelerations?
Thank you. And, you know, Lynne has been talking a lot about that agility to get product to market. I think a lot of people think if I'm agile to get product to market, I'm not building resiliency. And I truly disagree with that. I think resiliency and agility go hand in hand. Now, there are some things that we do that it's what I call the basics, that if you want to get product to market very fast, you do. You have a very solid development to commercialization tech transfer, if you will. You have a good data package that is very solid. You try to have manufacturing execution, right? And in manufacturing execution, I always highlight the data integrity piece, and we're going to talk a little bit more about data, but it is super duper important. And you also need to plan in an integrated manner, right? You have to think about the entire ecosystem when you're planning, about your suppliers, you have to think about your distribution process, et cetera. I think nowadays you have to add to those basics the collaboration with the regulatory teams. I think Lynne spoke about that. And you also have to talk about collaboration with the industry, partnerships within the industry. And you know, as she was talking about monoclonal antibodies and COVID, I work today at Lilly. My previous company was Amgen, and they partner to be able to bring product to the market very fast because Amgen had the capacity and the biotechnology expertise that could help Lilly ramp up very quickly. So we need to see each other as partners in a lot of the things that we want to do to be able to get product to the patients faster. Now, there are some things that are not as obvious when you think about agility and capacity. One of the things that everybody's like, oh, I know if I do that, I build resiliency, is just overall redundancy, having surge capacity in case you have some sort of variability or an unforeseen event, and also where do you locate that capacity. And that one is important because having all capacity in one place does not lead to resiliency. It could actually be very dangerous, and we've done that as an industry in the past as a way to reduce cost. So it's something that we have to think about. How do we locate the right capacity in the right places? Now, a couple of other things that are very important have to do with lifecycle management. How early do you think about that lifecycle management? Many times we launch a product, we have indications in our mind that we want to launch, but we don't really plan for that until the product is out there. And that's something that we need to plan ahead if we want to build resiliency and agility in the supply chain. We also need to think about scenario capabilities. And that one is more the business side of it, but trust me, when you launch a product, and because it's a new product, it might be a new therapy, you can experience all sort of volatility, both coming from the demand side, from your markets, but also coming from your supply and manufacturing. So it is very important to have that capability to build scenarios, and this is where data integrity that I was speaking to you about earlier becomes so critical. Because building the right analytics and the right models that can allow you to do some predictive scenario and some decision making, it's very, very critical. I think today a lot of people think about agility as how quickly you move the product. And there's a lot about agility that has to do with the data flow of the product and the decision making of the product. So we really need to build the systems for that. And I think one thing that we also don't think about as much, and this panel is talking to you about, is platforms. And you know, platforms seen as Anya was describing, the standardization of processes, the collection of technologies is very important for a supply chain. It is important for the science, the process, but it's also important for how do you build your company and your network. And it is how you get the economies of scale and potentially the technological advancements to be able to continue producing faster and in a more economical way for the patient.
CMC's responsibility, the supply chain teams, the manufacturing teams, it's all about that consistency and how are you assured of that consistency and control throughout the life cycle. Platforms is a topic that's come up, and Chetan, none better than you to talk about where it starts and the approach that you and your team have taken to establishing platforms.
Yeah, thanks, Anya. And why do platforms matter, right? I mean, Lynne mentioned that they were able to go from a sequence to a clinic in 5 months. There is 100 different ways you can get to that from point A to point B. And if you try to experiment all of those, it'll take you 5 years to get there. But when you have a platform, which is essentially standardization, you have a first intent process, which works and works very well. So when you have that context, which is built upon decades of experience, you essentially take the sequence and at the very first step decide, okay, which host cell line would I want to use that in? So you have a platform around a host cell and everything else that comes with it. You drop the sequence in and you get very predictable. It's not perfect, but you get very predictable outcomes very quickly. So that's a very simple example of at the very inception when you make a choice on which host cell line you want to use to express the antibody of interest. And then you can go a little bit downstream and say, how do I pick the best clone? Again, there you could do it 1,000 different ways. But if you have a platform that's worked for you, you could just drop it in that sequence and you end up with your lead clone very quickly. So there is speed, there's efficiency, but I would also argue there is reliability, which is sometimes an underappreciated dimension of having platforms. And it doesn't end there. Once you have your cell, you have to then put it in a process, express it, purify it, and so on and so forth. So the concept of a first intent process that works very well in most instances extends all the way through and even to formulation. So when you have something like that, you can achieve the speeds that Lynne talked about along with efficiency. Now, this may lead you to believe that platforms are very, boring and static. That's absolutely not true. If you go back to the first platform, maybe 25, 30 years ago, it looks super different compared to what is the standard now. I'd argue that platforms are very dynamic. In fact, we update them every year or so, so that you build in the latest improvements that you have. They're still a standardized first intent process, but they change. And when you move from platform A to platform A1, then your next molecule goes into that. One, feedback we got from our staff, Alison, you remember this, is, I've been trained to be innovative. And if I have to work on a platform, are you constraining my creativity? And the response was, no, bring the best innovation that you can, but take an outcomes-focused approach. If it's improving speed, if it's improving yield, that becomes a part of the updated platform. So they're not, innovation and platforms are not mutually exclusive. I would argue they're very tightly integrated, and in fact, platforms help focus innovation. And on the topic of extension, we have talked about antibodies, but many companies are now working on more complex bispecific molecules, where the principles that we have learned for antibodies apply. So a classic example is if you have an FC-based molecule, the first purification step, you can borrow a lot from antibodies and so forth. So there is that concept of extension, but there are limitations though. The formulation that makes an antibody stable will most likely not make a bispecific stable. So you have to go back and redesign your platform, if you will, for a complex modality. So again, I think platforms are not static, they're dynamic, they don't exclude innovation, and they are, I would say, you can translate learnings from one modality to the other. It's easier in express proteins, and as you move out, I think the principles will apply, but maybe not the specifics will start to change.
Thanks, Chetan. Peter, the third area that was mentioned, which enables the accelerations, are the regulatory pathways, and Lynne shared the news about the most recent guidance, which is applicable to COVID antibodies. You had mentioned previously the pilot program that the FDA is embarking on. I think it'd be helpful for maybe the broader audience to get a little bit more insight on that. as what it is and what's the intent of this particular program?
So we have a couple of different programs that are relevant in this space. One is essentially a CMC readiness pilot, which is trying to have products that are novel be able to be ready to go into production at scale by trying to address a lot of the issues up front. And so that's one of the things that we'll continue to work through. We also, in trying to leverage off of what happened during the pandemic, both CDER, Center for Drugs, and CBER are going to have a pilot program where for rare disease products, in our case in CBER, it'll be 3 gene therapies for rare pediatric diseases. In the case of CDER, it will be 3 products addressing neurodegenerative diseases, and where we will essentially give products the type of back and forth discussions that we had during the pandemic in their process of their development, hopefully overcoming a lot of the questions about product production early on. And I'll just use this opportunity to say that the other issue, just to clarify from a regulatory perspective, is that people may have heard that there is a platform therapies provision that was enacted into law last year. It indeed was. And the omnibus of 2023 put into place a platform technologies provision. That's different than a manufacturing platform. Manufacturing platforms could be a part of it, but this is really about platforms for products. And just to give you an idea of why this is so important, when we think about certain products, you can think about adenoviral vector gene therapies, or when you think about another platform, CRISPR, the CRISPR-Cas9 genome editing products may have more in common with each other than one might think, right? And most of the product will be the same except for the guide. And so this concept of platforms and being able to leverage will be very important, because for CRISPR, there'll be a platform that might cover many different diseases, and each of the different diseases may be subplatforms. So I think this is it's an exciting place where hopefully we'll be able to leverage manufacturing information, other preclinical information to try to help move things forward more rapidly.
I know we at VIR actually leveraged that same guidance associated with our early studies for CMV, so it is viral vector modification, and we were able to, simplify the path to clinical study by having an umbrella filing. So that was definitely very much appreciated, and an acknowledgement that, again, that dialogue really helps simplify. I think this is a good point for us to kind of reflect on our 30 to 40-year history with monoclonal antibodies and proteins, and look at our newer, more complex modalities. No one better than Alison Moore to give us her perspective on what we have learned, what lessons have we learned, what have we applied to these newer modalities, and where are we headed, Alison? What's your thoughts?
Thank you, Anya. So yeah, it's been fantastic for me in my career to see the evolution of platforms in the way that Chetan has described, particularly applied initially to monoclonal antibodies and then subsequently to more sophisticated manipulations, often of the FC backbone or for bispecific entities. And what was super interesting for me was when I stepped into the allogeneic CAR T cell therapy space, as was discussed a little earlier today in a different context, what's very present is the translation of something that was transplant biology in its first instance into a regulated drug product. And not just autologous CAR T therapy, but aspiring towards allogeneic CAR T therapy meant that really offered for the CMC scientists the opportunity to marry the best of what we've learned from these other protein therapeutics with these new whole cell engineered medicines. So a simple example of where we kind of got to work is that the wave bag bioreactor is in very common use in larger companies. It's often part of the specification of a platform, of a production system, the scale-up for a monoclonal antibody. And a lot has been studied and observed about the use of those wave bags and how cells grow in those bags, how to monitor and control the environment of those cells. And so scientists and process engineers then working in the CAR-T space were able to translate the information from that kind of platform history and retool it to expand CAR-T cells. So that's an example of where the evolution of first generation platform technology can be reapplied in advanced modality space. However, what I would say is the totality of a production platform for new modalities like CAR-T modalities is still in its infancy. Chetan and Lynne and others have referenced the fact that it was a tremendous freedom when our regulators allowed us to refer to prior knowledge assets, to refer to historical evidence in support of the consistent operation and practice of certain unit ops. That was very powerful and allowed us to build the history of those platforms. Of course, that history doesn't really exist for some of these new modalities. We're building it as we speak. That just takes some time. The other feature of new modalities that I'd like to call out, and to really broaden this discussion about platforms, is oftentimes the process development community thinks about platforms in terms of the production part of the platform. That is the workhorse platform, the unit operations in a particular sequence that result in the production of the entity that you're interested in. But platform thinking can also be applied to analytical methods and supply chain, as Noemi has mentioned, and now in much more molecular dimensions, as we're having much more sophisticated molecular engineering in our advanced medicines. And particularly for some of the advanced cell and gene therapy entities, our knowledge of what is important, and this is what Lynne was saying in the first instance, is that it's not just process knowledge and the process platform, but it's product understanding that is really critical to enable the freedom of a platform to generate speed, to generate geographical access, and to generate yield improvement opportunities that allow lowering in cost. Really what's key there is understanding critical quality attributes. And certainly when I started working in monoclonal antibodies, our understanding of those protein critical quality attributes was low. And in our regulatory files, we would just describe, peak A on a chromatogram. Whereas, 20 years on, 25 years on, we can, with great precision, specify what are the quality attributes that relate mechanistically to mode of action, also what are important for purity or safety or potency. With whole cell therapies, this challenge is significant. And what we understand currently about critical quality attributes for whole cell therapies is still a big work in progress. And until we make further improvements in our understanding about what's really critical, we have reduced freedom, unfortunately. But as has already been stated, there's been decades of work to build the initial platforms. We are certainly benefiting from that knowledge, but we have a ways to go in the new modality space to really get there. But what's a great precedent is that we know with good science and time that we will get there.
So our understanding of disease biology, Alison, has really increased significantly. Our tools that we can use to assess the products have improved significantly, but maybe we've made our lives just that much more complicated with all of this information and data. I'd love to hear from Matt, because there was a lot of excitement last month with the first CRISPR technology approved for Vertex, and also Bluebird Bio's viral vector approach. What are your thoughts, Matt, on how complex our business of CMC has become and our understanding of the product attributes?
So thank you. Yes, thank you, Peter. It's super exciting on December 8th to see the first CRISPR product approved, commercially approved, a mere 10 years after the report by Genetic Champier and Doudna of an in vitro use of an engineered single guide RNA to make a predefined cut in a piece of DNA in a test tube. So in many ways, one could say, well, what do you mean acceleration? In a decade, you went from a laboratory discover across the bay to an approved therapy that in the US and the UK and likely in Europe. So that's pretty good. On the other hand, I think we still have a room to go. Now, I just want to say for the 5% of the audience who doesn't know what CMC means, this was me 6 years ago. It means chemistry, manufacturing, and controls. That sort of doesn't lend itself to what we're talking about. It's really what we're talking about is, as been mentioned, translation and a bridge from an idea of how to make a drug to actually making the drug. That's what CMC is about, and that's what these experts all have spent decades of their lives figuring out, is how do you make a drug? And that's what CMC means. When I first heard the term chemistry, manufacturing, and controls, I totally didn't even get what that section meant. So if you're like me 6 years ago, you're okay. So a mere decade is really fast. I mean, it's not quite 5 months, but on the other hand, you had a platform already. We had no idea how to translate a test tube discovery into a therapy for our patient. And yet it really wasn't built from scratch. The gene editing field had a decade under its belt about what were the both on and off target considerations we needed to think about. And so the CRISPR field got to build on zinc finger nucleases and tau effector nucleases and homing nucleases. The Vertex product was built on 2 decades worth of work from Bluebird Bio and others about how do you even pherese CD34 cells from sickle cell disease patients in a safe and effective way. How do you treat them with how do you engineer them outside the body and get them transplanted? How do you even do an autologous transplant? So there was a lot of knowledge that already went into incorporating this biochemical discovery. So I think there's a ton to be learned from the history of monoclonal antibodies, and this is a group, and I'm sure there are many others out there who, but I also think that cell and gene therapy is not the equivalent of monoclonal antibodies, because an allogeneic cell therapy is different than an autologous cell therapy, which is different than an AAV in vivo gene addition therapy, which is different than an AAV gene editing therapy, which is different than an LNP CRISPR editing therapy. And so I think it's going to be very interesting over the next half dozen years to see whether these categories fall out in and unto themselves, or are we going to be splitting them out, or are we going to be lumping them together? And my suspicion is there's going to be they're going to be split, because I just think they're different entities. Finally, I'll just say, and it's been circled around, we want to identify the key analytic attributes, what was called the critical quality attributes, in our drug product, in our medicine, that predicts the clinical efficacy and safety. So we, I think, don't really know exactly what those are. And I'll pick on the CAR T world. We use leukapheresis packs that contain B cells and a heterogeneous mixture of T cells and all sorts of other mononuclear cells. It gets manufactured with a viral vector in general, and then it gets infused into a patient. We don't know whether the right, and there's a lot of hypotheses. Is it VCN? Is it the site? We don't even look at the site of insertions. We sort of look at the composition of the T cells, but we sort of don't. So we don't yet really know the critical quality attributes that allow us to say, that's the attribute that if I could reproduce it with a new manufacturing process, that's going to work just as well and just as safely in patients. And so we're really, I think, at the beginning of trying to figure those things out. So that relationship between the CMC people, and sometimes I wear a clinical hat, the clinicians saying, that product worked well in that patient. What was it about that product that predicted that, or was it just the patient that allowed us to predict? So figuring that all out is something we need to do. So I'll stop there so we have some time for discussion. Peter.
Yeah, no, Matt, I think you really, you touched on something that's it's something that we sometimes overlook about the fact that these are very different. And sometimes monoclonals get lumped in there with these, why isn't gene therapy just going to be like monoclonal manufacturing? And just to step back for a moment, the business model is completely different. Monoclonals, even the monoclonals for COVID-19, they're meant to be made on an ongoing basis with a business model such that you actually have a platform that works, right? You're going to make them on an ongoing basis because even though someone might receive them only for a year, there'll be someone else who will be receiving them for a year. The problem with the gene therapy and cell therapy model is that you're often doing, these are one-off treatments, often very different in a field that's very rapidly evolving with a device more, it's the evolution in this field is more like devices than it is like traditional drugs. So it's a real challenge. And then I think we've seen the fact that many of these have very unique manufacturing considerations that have not let us settle on something that's as simple as the monoclonal platform. Now, if I were to hazard in my crystal ball, we have a bridge time over the next 5 to 10 years where we're going to be dealing with kind of individual manufacturing platforms. But I suspect once if I looked at my crystal ball, I see CRISPR inside an LNP targeted by an antibody, where that's going to be a platform where we might get back to a monoclonal-like manufacturing process. So I think we have a bridge period over the next 5 or 10 years till we catch up and are able to move to what may be a much more facile manufacturing platform. The question is, will we be able to handle the cost and the complexity over that time without having too many companies go belly up?
Yep.
How do we take this and recognize that that future is very complex? How do we ensure that we're working better together, both with the agency and with each other, to overcome some of those complexities? Because as you said, Peter, no one company is going to have so much experience that we can truly, have a platform approach within a company.
So I would say from my perspective, there's a fair amount of dialogue necessary here. I think it probably will help to have more dialogue about both, and I think it's two types of dialogue between individual companies and the agency, but also dialogue among companies, because there are certain best practices that hopefully we could agree upon that would really be helpful. I mean, even simple things like having agreement on kind of how we go about making AAV vectors in academic labs so they can transfer easily to CDMOs and companies with uniform protocols. We haven't been able to achieve that, and if we could, it might be helpful. So having those dialogues about what we could achieve by getting there with, collaborating with industry and academics might be very helpful.
I was really excited this morning to hear from Robert Califf the opportunities at the agency to work between divisions as well, Peter. And so that's, I think, something that is going to really come to life in the coming years. But certainly an industry-type collaboration where we can bring those experiences and work with the FDA through we have the Emerging Technology Program. Is that the best mechanism for these types of dialogues?
Yeah, I think the emerging technology teams, both ours at CBER, and in some cases, some may have ones that there's a similar team at CDER, may be very helpful. I also wouldn't neglect that we probably should bring academics into the loop. It really does help for academics to be able to peer over and see what, over the kind of the fence and see what CMC looks like, because then they may better understand why it's really necessary to try to do things in a standardized manner so that we don't have to reinvent the wheel every time, because deriving a cell line anew from a company costs the company a tremendous amount of money, but it costs patients time because it delays any product by 6 to 18 months. I mean, someone can comment on that. I mean, I'm sure. I mean, when I was in industry, we made the mistake of buying a product once, acquiring a product that had to have a cell line derivation, which ended up taking 18 months, and it unfortunately made the net present value calculation of that acquisition negative.
And I know, Lynn, if I can just build on what Peter said, though, I think it's really valuable for academics to look into what biotech and big pharma CMC looks like. As I said, I've learned a ton in the last half dozen years by doing that. On the other hand, there's another advantage to it, which is I can look into what they're doing and go, that's crazy. Why are you doing it that way? That's so complicated, and allows then academics, the engine of innovation in academics, to start solving these process engineering problems.
Yeah, and it would be, Matt, I really like that, because it would be really wonderful to stimulate people to actually, I mean, academics usually when you start to talk about CMC, it's time for Z and Z. Sorry.
Maybe we have time for one audience question. Is there anyone that would like to pose a question? Any of our academics have any advice or feedback? I think we're close to out of time, but Kathy, or...
I just wanted to comment that although, I'll speak without although this is for manufacturing, I think that the ideas of creating platforms to create an efficiency, to account for the efficiency of manufacturing can be taken into the clinical setting and paired with platform trials, that we actually need, that the whole idea of trials like CASE-by-2 and others, the whole idea was to take these concepts in efficiency in manufacturing and bring them into the clinical trial arena, and that same flexibility is also needed to continue to build the platform where you don't have to start over every time. So I think there's a lot that we can learn and share from the manufacturing process to the idea of how we are testing trials. And I think a lot of people from the industry want to have their own trial and do things, but it's incredibly inefficient.
Yeah.
And that I would recommend, I want to see what you think, that that's also a way to help you advance what you're doing, test it more rapidly, and keep going.
I think Noemi has some great examples on that one.
Well, actually, just going to you're right on target, by the way, Laura. Thank you. Thank you for that comment/question. When I worked in clinical supply chain, they used to joke with, when you take a product to the clinical trial, you build 1,000 supply chains that you use one time. Yet in commercial, typically you build one supply chain that you use 1,000 times. The reality is that in the commercial world, we're no longer building one supply chain. We're building many, right? And I think the key here is how do we balance out, and I think that was the purpose of this panel, to talk about platforms in that perspective. How do we take what we know? How do we connect the data that we have so we can create that efficiency so we don't have to create 1,000 supply chains for the patient?
Yeah, and one particular advantage is you have one large database where you
Correct.
actually can compare, and that's in everybody's interest.
Correct.
Even though people want it, like we need to do to more pre-competitive sharing, because that actually helps everybody.
I think Chetan has a comment as well to build on that, Laura.
I'll just make a philosophical comment, right? Most creative people are not wired to think that way. I completely agree with what you said, but it's very, very difficult to tell people to standardize and follow a standard process. So we just need to kind of work with that, and it's going to take some effort to really expand. Those of us who have done it and lived it, we appreciate it, but I think it's very hard actually to kind of get that across folks who have not. And then maybe to to pick up on Peter's comment, that in our 45-year history, we have changed the cell line 3 times, right? So I think that just speaks to, one, it takes a decade of work to bring about an improvement. And once you do, you stick with that, and then with the next decade, you make a change. So it's a pretty, you just can't make those changes, that frequently. But when you have one that really works, it makes a tremendous amount of difference.
I just wanted to say one real quick thing, that we've been using the term platform, but really it's about doing the hard work across all disciplines, the convergent thinking to get to simplification. And that's on the regulatory side and on the scientific side. So the accumulation of regulation works against us sometimes, but if we can bring new thinking scientifically and new thinking in terms of regulatory strategy, and together really try to find the simplest route and keep iterating on that, that's what allows us to create the future of medicine.
We've one question from the floor. I'd love to make it.
Thank you, everyone, for your time today. I guess the question to the panel is, what role do you think technology can really play in trying to bring together this platform knowledge that exists within a modality, within a company, at the regulatory level, where they've seen a lot of applications, and actually giving it back to industry, across the wide experience that the agency has over several different drugs that they review from a CMC perspective? And, how quickly could we get there, and what does technology really do there?
I think what we agreed as a team collectively as we were meeting to prepare for this panel is that we need to continue the dialogue. We need to strengthen our partnerships with the academic groups. And Laura, you were very firm in your request for a commitment or a call to action. And I think that's ours, is to continue to work together, work with Peter and his reviewers, and use this opportunity of this general broad knowledge across so many companies and so many modalities, and really try and understand the challenges and see what we can do to simplify, accelerate, and ultimately deliver these medicines to patients. So thank you, everybody, and thank you to the panel today. It's been a lot of fun, and appreciate it. Yeah.